Just another few seconds. All right. For all those people that are here on time, we really appreciate you. So we want to go ahead and get started. Hello, good afternoon, good morning, good evening, depending on where you're from. But my name is Morri Chowaiki, and I'm the Chief Sales Officer here at Viome. And welcome to the fourth webinar that we're hosting called Signal in the Noise, and where we really try to decipher your Viome results and recommendation, give you lots of insights into our company. And today I have the distinct pleasure of having a conversation that I'm really looking forward to with our Chief Science Officer, Dr. Momo Vuyisich. Vuyisich, did I say that right, Momo?
Close enough.
All right. Well, I want to, it's a real pleasure to have this opportunity today. And Momo is one of our co-founders and Chief Science Officer of Viome and Viome Life Sciences. And Momo's one of the smartest individuals I've ever met. You know, we spent some time together, and he really makes me feel like my 30 years in the life science industry has been a complete waste of time because he's so brilliant. Momo, I'm going to put you on the spot. Can you tell us a little bit about your technical background, your qualifications, and just, how you got into this world?
Sure. Technical background. So I have an undergraduate degree in microbiology and almost chemistry, one class short of a double major. And I have a PhD in biochemistry. And then I have a pretty long academic career of writing proposals and grants and doing research in academia and the government. I was sort of a jack of all trades. I tried to solve many problems in parallel. So I delved into every kind of a science that you can possibly imagine: environmental science and cell biology and virology and immunology and cancer biology and everything. So really wanted to develop sort of a broad spectrum of technologies that can study the whole planet as an ecosystem. And in fact, the technology that Viome is currently sitting on was developed at Los Alamos National Lab. And in my grants, when I was seeking money to develop this technology, the constant theme was that this technology can be used for planetary health, which is we can use it not just to study the human body, but also any kind of an ecosystem, whether we're talking about the ocean ecosystem, coral reefs, or animal health or plant health or soil health. Even we can use it to study, the health of space systems when we move out of Earth and so on. So that's sort of a little taste of my career.
Amazing. Well, can't wait. And, I'm looking forward to today because, I really, this is meant to be a conversation. We got a couple of graphics that I'm sure we're going to pull up, but this is really meant to be a conversation. And, regardless of the hundreds and hundreds of people that we've got watching right now and that will watch later, this is actually a kick for me because I get to know you better. And I'm really excited to really understand how this all started, since I've only been here a year. But, some of the things that we're going to touch on today is something that you call Nutrition 2.0, which I can't wait to hear what the heck that's all about. I'm going to ask you, kind of about your health journey. And, from what I hear, the rumors are that there's something really personal that kind of got you to where you are with Viome and why you started it and why, and how Viome was created. And then, you're in the lab, you're in the white coat, you're there day in and day out. I want to really understand for our listeners, what is the journey of a sample? Once they send it in, like what actually happens and how do they, from the time we receive it to the time that they get their results, I really want to understand and hear it from you. And you have this amazing ability to talk at a PhD level, but for somebody simple like me to understand, I want to really understand the difference that we always talk about between RNA and DNA and, about our research and all the improved diagnostics and therapeutics that are coming out every single day. So Momo, if it's good with you, I'd love to get started, unless there's anything else you want to tell everybody, like, what sign are you and what's your favorite food?
Let's do a spoiler alert. DNA is boring and RNA is sexy.
Wow.
More to come.
Mic drop. All right. Well, with that said, Momo, let's go ahead and get started.
Right.
First thing I've got to find out, because this is a little teaser that I got, Nutrition 2.0. What on earth is that all about, and why is it important?
Yeah, so Nutrition 2.0 is just a sort of a standard computer jargon for an improved version of the current nutrition. So nutrition really over the last few decades has focused on nutritional deficiencies, meaning a nutritionist can test you for whether you have low iron levels. And if you do, they'll recommend that you consume more iron, right? And also nutrition has really focused on fad diets, and that's really a sad and frustrating story because I was a victim of these fads, and it was a very frustrating time in my life. And so, if you go to three nutritionists and you give them the exact same story about what symptoms you're feeling or what you would like to improve, you're going to get three different stories. And these are supposed to be nutritional scientists. How, if you ask three mathematicians, how much is 3 plus 3? You're guaranteed to be given the exact same answer. So why is nutritional science not really scientific? Why is it all based on people's opinions? And so that's Nutrition 1.0. And one of the foundational principles at Viome is to leave all of that subjectivity behind and all that, basically nutritional religion behind, and to really go after a very objective science. And so Nutrition 2.0, in simplest terms, can be defined. Oh yeah, we have a slide for that. Here we go. So you can see the Nutrition 1.0 on the left side is addresses nutrient deficiencies, which doesn't always work, promotes a fad diet that is best for everyone, and then creates mass confusion, which is not great at all. So Nutrition 2.0, these are the three main aspects, is that it is 100% data driven. And what that means is that today, when someone submits a sample to Viome, the sample goes through the chemistry laboratory where we measure the chemistry, meaning instruments measure the chemistry of what's going on in those samples. Then mathematical equations overlay on top of those chemistry data in order to compute everyone's recommendations and everyone's scores and everyone's supplement recommendations. So that process is 100% objective. Now, I'm not saying that it's 100% accurate yet, but it is 100% objective. There is no human influence whatsoever. And what that means is that as we're gathering more data, that process that is 100% objective will keep improving until it reaches nearly 100% accuracy. And at that point in time, it'll be 100% just chemistry and math that is 100% accurate. And that's really what we're doing at Viome and what we're trying to achieve. So what this means is that because everyone's chemistry is different, that means that everyone's going to get a different recommendation. And so our computers don't really know anything about Mediterranean diet or ketogenic diet or any of those diets. They simply compute exactly what ingredients a person needs to consume and which ingredients they should avoid, and then maps those to the foods and supplements. And really, the ultimate goal of Viome is to use specifically nutrition to prevent chronic diseases and cancers and to slow down aging. And how we're going to do that is simple. When you're in your 20s and you're super healthy and you're running triathlons and so on, that is a physiological state of health where you're physically, mentally, and cognitively very healthy. Why can't we just maintain that state? Well, we can. We just need to learn how. So if you think of a human body as like a really, really complex machine, like a twin turbine helicopter or an airliner, they can run for decades absolutely perfectly. All that we have to do is prevent problems. We don't fix problems on airliners in the middle of the flight. We don't wait until they break down and then fix them. We have preventative maintenance for those complex airplanes so that they're always perfect, so that they don't break down in flight. That's why they're so safe. And that's exactly the metaphor for what we want to bring to the human body. We just have to measure the right things. We have to measure the right molecules. And that's what Viome's platform does. We measure RNA, and more on that when we get to that stage.
And we really haven't had access to this type of technology in the past, have we, Momo?
Exactly. That's the main problem, is that in the past, other technologies have looked at the wrong things, which is DNA, and DNA is static and boring, and we'll expand upon that. Yes.
So you must have had a bit of an aha moment to come up with this. I mean, however long ago it was where you developed this, and I think you were working for the government at the time at Los Alamos National Lab. There must have been something that was going on with you. I mean, most entrepreneurs have, something personal, something happens to kind of, to take the initiative to really make a moonshot or a game changer. So,
Yeah.
Was there anything that was going on with you personally that, you were experiencing that you said there has to be a better way, or I can't find the solution, so I'm going to make it? What was the inspiration for this?
Yeah. Yeah, there are a couple of inspirations, a professional and a personal. And so, yeah, I was a scientist and doing what scientists do, which is do experiments and publish papers. But I started to realize, even in my PhD career, that that sort of spinning wheels for the rest of my career. And I realized that I was going to retire with what did you accomplish in your life? Oh, I published 120 papers, and that's it. That's your one shot at life. What do those papers actually do? Do they actually do anything? Well, they do very little. So early on in my professional career, I realized that we have a huge problem with chronic health and healthcare system in the United States. And there are two entities that people think of as pushing healthcare forward. But in fact, if you dig a little deeper, you realize that academia, this is a system with hundreds of thousands of super smart people, well-funded, hardworking, cutting edge science, yet they're incentivized to publish papers. They're not incentivized to come up with a cure for multiple sclerosis. No one is incentivized. They don't get paid more and they don't get more grants if they find a cure for multiple sclerosis. They need to publish papers. So I realized early on the purpose of the main purpose of academic work is to publish papers, no matter what that is and how relevant it is. And so that's a real shame. The second, you look at pharmaceutical companies, and pharmaceutical companies have never come up with a model, a business model that benefits from people being healthy. That doesn't exist, right? So everyone benefits only if they keep you sick for the rest of your life and manage your illness. And so a long time ago, I realized, wait a minute, there is no entity in the world that's actually using all of that scientific knowledge and technology to actually come up with a preventative strategy. There are obviously thousands of conferences, there are thousands of meetings, there are many, speeches that people give in terms of how important it is that we introduce prevention and how critical the need is and how currently that need is not met. There's tons of talk, but no one has said, wait a minute, someone actually needs to do something about it. And so I set out on that journey to do that. And that was driven in a big part because I developed a chronic disease early on. So in my grad school days, early on, I developed something that no one ever knew at that time what it was. Some doctors thought it was rheumatoid arthritis. Others thought it was ankylosing spondylitis. Some doctors simply labeled it as autoimmune because they didn't know what it was. And that seems to be still 20 years later. When a doctor says it's autoimmune, that likely means they don't know what you have. It's just that they know you have inflammation. They're going to pour some water onto your coals and try to cool it down. And that's the state of the art. So I developed that disease and it was progressive. And so that means that, 10 years later, my joints started to lock up and I was in pain 24/7. I was on painkillers 24/7 and I was in my mid 30s. And that was obviously a very bad situation to be in. And I was sure that there is something wrong with the diet and nutrition. And so I tried all the fad diets. So I spent a couple of 3 years trying these different diets. And if you're guessing, you have no idea what you're doing. And so my disease was going through ups and downs. And sometimes you start a diet, and then the first week you start to feel better, and you're like, "Wow, this might be it." But then you get worse, then you get really worse, and then you're
You
like,
think that was a placebo effect?
"Yeah, exactly." You just don't know. And interestingly, and I'll make a point out this later, I even tried a vegan diet, but I only tried it for 2 weeks, and that was the problem, unfortunately. And at that time, a vegan diet meant salads. And after 2 weeks, my wife and I said, this is not sustainable. Like, we can't live like this. And because I wasn't improving in 2 weeks, I said, well, fine, let's just quit it, right? And so tried all the diets until I tried the ketogenic diet. And after 3 months on the ketogenic diet, I actually started losing memory. And it was, I started losing it slowly. And so it didn't really click or anything. I thought something, I'm just getting old, whatever. But then after 3 months, it was really noticeable and pretty severe. And I thought, wow, this could be induced by the diet. And so that was many, many years ago. And I still cannot say that I'm 100% recovered from that, although I'm mostly recovered from that. But it was an incredibly bad experience. And so I stopped fad diets after the keto diet. And now I know why the ketogenic diet induced what it induced, but I didn't know at that time. So this story ends with, I found a study that identified a small molecule that is produced by all mammals except humans. And that means that all mammalian foods, such as red meats and dairy, contain this molecule. It's called NEU5GC, NEU5GC. And the studies determined that it was likely the cause of chronic inflammation, or a major source of chronic inflammation in humans. And they actually showed in mice that it causes inflammation and cancer when they made a mouse system that was similar to humans. And so I read those studies. I was willing to try it. And so one day I simply quit all mammalian products in my diet, 100% clean. I knew the biochemistry of this molecule, and I knew that it would linger in my body for quite a long time, and I couldn't have cheat days, and I had to stick with it for at least 2 months. And after a month, I started steadily getting better. After 2 months, it was absolutely clear that this was the cause of my problem. And so because my body was in a pretty bad shape, it took about a year to heal. But since then, I've not had any problems. My disease was completely cured. And so identifying that antigen that was found in mammalian foods was the key for me. And that was really a big stimulant to shift my scientific focus, develop the technology, and my teams spent 6 years developing the technology that we use today at Viome before we started Viome, to make sure that we're actually that we're using a technology that's relevant to the human body and diseases, and that it's robust and it's automatable and it's scalable and it's cheap enough, because at that time it wasn't cheap enough. It was absolutely not going to be a consumer product. All right, that was a bit of a long story, wasn't it?
No, but that kind of insight and transparency is really appreciated, Momo. Thank you for sharing that. So you developed this technology. You're using it for other means, and like when did you meet Naveen, Jane, our founder? What was kind of going on at that time in the industry? What was the light bulb, and how was, tell me a little bit about like how Viome was created. I'm fascinated. I've never really understood the story, and I can't wait to hear it, and you to share this with our listeners too.
Yeah, we have a slide for that, and it's a
I love it.
And Shark Tank really well, of course.
I do.
Yeah, so Morri, I watched your Shark Tank presentation. Super smooth and creative. Loved it.
Thank you.
My teams worked for 6 years to develop the technology and get it to the point where it was robust, it was reproducible, it was scalable, it was automatable, and it was cheap. That was one of the biggest problems with metatranscriptomics. And so that year, 2016, I was looking to commercialize it, and Naveen came with his investment company, and we had a Shark Tank at Los Alamos National Laboratory. And short story is, I won, right? So, I had a pretty good business plan. The technology was very ready for commercialization, and so Naveen and I started Viome back in 2016. And so that's a picture of us in the middle right before we started Viome. And then
Momo, I see your desk over there. I think I'm going to ask Naveen for a little bit more budget to get you, a proper chair and desk, if you don't mind.
Sure. How things have changed. So that was literally that was the first day in the office after starting Viome. We moved into a facility that eventually became our lab facility, but it was just an office facility with carpets and no furniture, and you start from nothing. And so my desk was 2 trash can boxes. That was the first day. But that's how you start a company. You started from nothing. And I was fortunate enough to bring some of the brightest members of my team from Los Alamos National Lab to Viome, and we hit the ground running fairly quickly. And 5 months later, we were starting to package boxes and ship them to customers. So that was pretty awesome.
That's amazing. So, kind of what was going on with healthcare at the time, and the chronic diseases, like how was besides kind of the idea, what are some other areas that you can kind of fill us in on how was Viome created? How did it transition to the Viome Life Sciences and the platform and the applications? Give us a little bit of insight there, if you don't mind.
Yeah, it's a bit of a long story, but in summary, the platform that we created at Los Alamos National Laboratory is basically a systems biology platform that can take any sample, any human sample, any soil sample, any water sample, any air filter sample, any animal sample. I mean, literally anything you can imagine. And and process it in a universal way, which is we want to extract all of the RNA from that sample, which is the gene expression of every single microorganism and every organism in that sample, and understand the gene expression profile. And we'll get to that a little bit downstream. It gets a little technical, but that technology then can be used for any kind of an application. So can we go to the next slide where I think it'll be nice to visually show. So I would like people to think of Viome Life Sciences as that's now the legal entity that owns Viome. And so Viome Life Sciences is the owner and maintainer of what we call the ViOS platform. So ViOS is similar to iOS, but it's Viome operating system. And ViOS is a systems biology, it's a technology platform similar to the App Store. So the App Store consists of iDevices and software, and our ViOS also consists of laboratory hardware, supercomputers, and software. And ViOS platform enables us to collect samples from anywhere in the world, analyze them in our labs, and deliver results recommendations to customers, but also learn from those data and understand systems such as the human body in a much better way to understand what is healthy, what is sick. And then most importantly, not only just tell someone, hey, you're sick, they already know that, but tell them, hey, you might get sick in the future, or you're likely to get sick in the future. And then specifically, here's what exactly you need to do in order to prevent that. That's really the key for human health. So think of Viome Life Sciences as a parent company that owns this platform. And just like the App Store, our platform is completely open access, meaning that 100% of all the technology that I have access to, every person on the planet has access to. So we have a grants program, people can apply on our website, and we've already given grants to other institutions. And we already have one application that we're developing with the co-partner at Queensland University of Technology. We currently have an actual clinical research program with University of New South Wales, several pharma companies, some other universities, and smaller hospitals, all via this open access program. And so you can see on this slide, what I'm showing here is that box on the bottom is ViOS and all the components, but really the applications, just like the apps on your phone that we're developing, are all listed here in the currently what we're working on, and then short term and then long term. And so I want everyone to envision Viome 10 years from now to be a systems biology company that has spun out 1,000 health applications, where some of those applications were developed completely by Viome, such as Viome.com, and then other applications that were developed with partners, where the partner maybe did most of the work, but we gave them turnkey access to our technology, and they offer a product that has a certain clinical utility or lifestyle utility, and they're offering it and they're selling it, but we just get royalties from them. So that's a business model where we can exponentially amplify the potential of this platform. Instead of waiting 100 years for Viome to develop all applications, we're inviting the whole world to use our technology and to develop their own health applications so that we can benefit the most people most quickly.
Amazing. So I want to ask a question that kind of fascinates me. You know, when I started, I've been involved in the vitamin industry for 25 years. And when I started working at a large manufacturing company, the first question I asked is, I've always wanted to know, how does that little tiny pill get in the bottle? Like, I want to know all the way from the beginning, huge bag of powder, how does it work? And that was the most fascinating thing for me.
Yeah.
So kind of the same thing I want to ask you here, Momo, is, you know, I know our technology is amazing, and, you know, we've got, you know, something that's totally proprietary on what we use to process the sample and formulate the results. But do me a favor, can you walk me through the journey from the time a customer sends in their sample and our lab receives it? What happens to that sample? What is the chain of events from that time to the time they get their results in their app?
Yeah. Okay. So I'll try to compress this in a few minutes, but let me first give you an example. So when you get prescribed to get a lipid panel test by a lab, by a physician, you'll go to a lab, you'll get your blood drawn, and they want to measure cholesterol. So cholesterol is a molecule. In chemistry, we call it an analyte, but it's a molecule. It's a physical molecule that's found in blood. And the lab wants to understand what is the level of that molecule or the concentration of that molecule in the blood. So they will use some kind of an analytical method or instrument to measure the concentration of that molecule, and they will report it in a digital form in some kind of a PDF document, right? And so we follow the same principle. Every lab follows the same principle, which is we take a physical sample, we analyze it, and we digitize it. And so the purpose of our CLIA lab is to convert physical objects into digital data.
What is, Momo, before you go on, what do you mean by CLIA lab?
Okay, yeah, let's talk about that a little bit. So one of the unique features of our laboratory is that we are CLIA licensed. So that means that the CLIA division of HHS, the US federal agency, oversees all of our operations.
And CLIA is CLIA, right?
Yeah, CLIA is CLIA, yes. So it's a clinical laboratory standard. If you want a physician to order a test, and if you want a physician to act upon a test, it cannot be a research test. It has to be a clinical grade test. And so CLIA is a system that includes the test itself, but it also requires 1,000 different things to ensure that the test is performed properly. And so think about this. If you were to want to do an HIV test to see if you're HIV positive, and you go give some lab a test, and you get a positive test, and you're like, wait a minute, there's no possible way that I'm HIV positive. How sure are you that I'm HIV positive? And they say, well, you know what? We had some reagents thaw yesterday, and there's this guy who processed them. He's just not really good in the lab. So I don't really know if I trust the data, right? What would you do with that information? You'd say, well, this is sort of a useless test. I don't really know if I have no idea if I trust it or not, right? Well, in a CLIA lab, when you get that result, you know that it's the right result because every single instrument has to be qualified and has to be maintained and has to be continuously assured that it actually performs to a certain standard. Every single reagent has to be properly stored, maintained, and qualified. You can't just buy a reagent from a company and say, well, that's a good company, I trust it. No, you have to qualify it. You have to spend time and money in your own lab to qualify that reagent to ensure the performance. You have to do environmental monitoring of everything. Every single technician in the lab has to have college education, has to be properly trained and competency tested. So the list goes on and on and on. But the ultimate goal of the CLIA licensed laboratory is that when they produce a result, that result can be trusted instead of coming up with 1,000 excuses as to why that result may not be accurate. And I did not want to leave my cushy government scientist job until we reached the level of performance of this test that it could be CLIA certified, because I didn't want to do research in a business setting. I wanted to scale with a very robust test. So that was we did sort of a CLIA validation prior to starting Viome, a mock CLIA validation, and everything worked perfectly. And so that was a big test. Does that answer your question pretty well?
It does. Thank you so much. Is there more that you want to reiterate on the journey of the sample?
I mean, yeah, there's a lot to it, and it sort of gets technical. But the point is, let me get back to lipidomics. So you go to a clinic, and they draw 5-10 mils of your blood, and they give you 4 data points: HDL, LDL, cholesterol, and triglycerides, right? They measured 4 different things. Our blood test produces 15,000 data points, and that's just the start. We're actually going to produce a lot more as we develop bioinformatic analyses, but we get 15,000 data points from finger prick blood. And that's really the power of Viome, is that we're getting—
Wait a minute. When I do a lipid panel, they're pulling like 3 tubes. We're providing 4 drops of blood.
Exactly. So that's a dramatic difference in the sample size that we were able to work out, but also we're getting 15,000 data points instead of 4. And then most importantly, we're getting the 15,000 data points that matter. And we're going to definitely go touch upon why RNA is so important and why DNA has basically failed on its promise and is not really that useful. And I have some nice slides to show you. So the journey basically, stool and blood and soon saliva, they're all received in the lab. They're accessioned, which means they're scanned, their barcode is scanned into a LIMS system. It's a software that recognizes samples and makes sure that it knows where the sample came from and that it's due for analysis. And then the samples are handed to our CLIA lab, where they're processed using liquid handling systems that are fully automated. So our technicians don't even need to know how to pipette, because everything is fully automated. And in that process that takes now about 7 hours, what happens is that we break open all microorganisms in the sample, regardless of what sample it is, whether it's stool, blood, or saliva. In blood, we obviously break open all human cells as well. And then all of those contents of those cells are then purified. And then we remove some of those, very few of those we remove because we don't want to just keep sequencing them because they're boring. We don't like to work on boring things. And then we sequence everything else. And the sequencing means that we're reading the sequence of A's, C's, G's, and U's in RNA, slightly different from DNA. But we get that sequence and we quantify every single molecule, just like you would for cholesterol, on a sequencing machine, so that at the end of that process, literally what leaves our lab are digital data that go up in the cloud, massive amounts, of course. So that's a summary, I would say.
All right, Momo, you had me at sexy. You started this conversation with a spoiler alert about DNA being boring and RNA being sexy. There's all these gut microbiome tests out there, including ours. Why would somebody pick Viome over the rest of them? And does it have to do with the sexiness of RNA?
Yeah. All right. Well, I'll let you make the sales and why they should pick our tests over others, but I really want to show a couple of slides to show how dramatic the difference between RNA and DNA is. So,
But are the others, just so I understand, are the others looking at DNA and we're looking at RNA?
That's correct. We're the only test that performs RNA sequencing for gut microbiome or oral microbiome or blood. Everyone else is focused on DNA. DNA was so hyped up back in 1990s, and after the Human Genome Project finished, the promise was made by the scientific community that once we sequence the human genome, all chronic diseases will go away. What they completely missed was that the root causes of chronic disease is not our genetics. It's our gut microbiome, oral microbiome, and our nutrition and environment. It's not the genes. And that's clearly now demonstrated in peer-reviewed publications. But they just didn't know that. And some people knew it, but they were not loud enough. And even today, we are struggling to be loud enough with RNA versus DNA because the world is so ingrained in DNA. And now, the nice thing about DNA is that it's easy to work on. There's plenty of data. You can publish papers. And so everyone is still very much focused on DNA. And RNA is very, very difficult to work with. It's very difficult to work with, So let's go with this graphical representation. So most people don't actually realize that every cell in their body has the same exact DNA content. So if you were to sequence the genome of a kidney, liver, and brain cells, those genomes would be identical. So if I gave you a sample and I asked you, hey, if you sequence by DNA, can you tell the difference between these three tissues or organs? You'd say, well, I can't. They all have the same DNA. That means that the genome you inherited from your parents has the potential to be any of the organs in your body and has the potential to be healthy or sick, but it's all a potential. That's it. There's nothing more to it. And so if you sequence these three tissues by DNA, the same result. If you sequence these three tissues by RNA, every one of those tissues has only expressed or activated a fraction of the 22,000 genes that the genome encodes. So a few thousand genes are expressed in each one of those tissues, and they're very different. And it's the expression of those genes that then determines not only the shape and the look of these tissues, but more importantly, the function. So that's really a critical thing for people to take away, is that these tissues are vastly different from each other, not because they're genes, but they're gene expression. The second thing is, now we go into the next level, which is really important. Let's go to the next slide. Not only is RNA absolutely critical for distinguishing different tissues, which is sort of potentially an academic thing, but it's extremely important to understand that the difference between health and disease is not in the DNA. When someone gets sick, the day that they got diagnosed, the day that someone develops the symptoms of multiple sclerosis or Alzheimer's or diabetes or obesity or any disease, it's not like their DNA changed. It's the same DNA they've had since they were born. It's that their RNA expression or gene expression profile has changed. And that's demonstrated very, very nicely here. So in an IBD model, so this is Crohn's disease model, where you have Crohn's disease patients going through periods of relapse and remission. One day they're healthy, next month they're sick as a dog, 3 months later they're healthy, they go again through phases of relapse and remission. Their DNA is the same. their microorganisms in their gut microbiome are essentially the same. They're all there. They just shifted a little bit in terms of composition. So you can analyze this system during the periods of relapse and remission using DNA, and it's going to return the same result. It doesn't change. What changes absolutely dramatically, both on the human side and the microbial side, is the RNA. And that's shown here in the picture on the left side, where you have the same exact small intestine on the left side in the remission, and that same intestine a month later in relapse, you can see that they're dramatically different. And that's because of the RNA expression. And the graph on the right is a heat map of a gene expression profile that shows those differences in RNA expression. It gets a little technical, but there's really solid evidence. The same thing happens to the microbiome. It's not like, oh, my microbiome is gone, I replaced it with a new microbiome, and that's why I now have a relapse. And then you somehow magically replace your microbiome with a healthy one, and that's why you go into remission. It's the same microbiome you have, it just changes its functions. And the functions of the microbiome and then downstream the human genes are changed with the ingredients in the foods that we eat. And so now we have a system where we now know that nutritional ingredients can actually modulate our physiology either directly or via the microbiome. And that's a really fundamental concept of Viome that we're pursuing every single day. Does that make sense, Morri?
Makes perfect sense. And what's to stop some of our competitors from sequencing RNA?
Well, there's nothing to stop anyone from doing anything, first of all. Second of all, I don't feel like we have competitors or we will have competitors. We need to revolutionize healthcare. We need to change a massive, massive system that is currently a sick care system. And we can do that alone, or we can do it with other so-called competitors. It doesn't matter. We want to improve humanity. So even if competitors were to do this, it's fine. But we developed this technology 7 years ago and patented it at Los Alamos National Lab. No one yet has come out with this technology. 23andMe tried and failed. Google tried and failed. It is very, very challenging. I was lucky enough to have the background in RNA from my early science days, so back to mid-90s. And I was gritty enough, I was persistent enough to know that I can make this work. And so I persisted and I never gave up until it worked.
Thank goodness you're stubborn.
I call it grit, Morri. Grit. Stubborn is sort of a bad word. Grit. Never give up.
And listen, I know we've all got that mission, and there's obviously our consumer division that is focused on our products and what we're doing outside and in health sciences is phenomenal. But I also know that we're doing, we've kind of got another arm, and that's research. And research has been a part of your background for a long, long time. So, we're doing a lot of clinical research. Tell everybody a little bit about what we're trying to accomplish by way of doing so.
Yeah, actually I will, but I want to show the audience one more slide, which I have on RNA versus DNA.
Sure.
Yeah, so I hope that people understand that RNA is dynamic and it's sexy and it's relevant. The most important keyword for takeaways is it's relevant. It's relevant to health and disease. And so on this slide, I'm showing, so one of the, for example, since you like to talk about competition, one of the only things that the competition can do with their DNA-based test is to tell you, hey, great news, you have Faecalibacterium prausnitzii, that's a great butyrate producer. What they're not telling you is that Faecalibacterium prausnitzii can produce butyrate, but it does not always produce butyrate. It depends on what you feed your microbiome. What they also don't tell you is that Faecalibacterium prausnitzii actually does not produce butyrate from the fibers, so you cannot just eat fiber and it'll produce butyrate. It's actually cross-feeding with other members of the microbiome. It requires some other metabolites from them. And so you have to understand the context of the microbiome. You can't just say you have Faecalibacterium prausnitzii. So I would like the audience to understand that just because you have a potentially beneficial bug detected by DNA, that's all it means, is that it has a potential to produce butyrate. But how do you make it produce butyrate? You have to feed it and its partners just the right ingredients and not feed it other ingredients, because if other ingredients are fed, they're going to prefer to feed on those and they're not going to produce butyrate. And so really you have to look at the whole community. microbial community, and you have to understand what ingredients you have to provide in order to make that community healthy for you. Because remember, when I went over the IBD example, the same community can be healthy and the same community can make you sick. The same community. It all depends on what diet you're feeding them. And so that's really a key takeaway, that all tests that are using DNA will only tell you about the potential. And that's it. They stop there. You don't know whether that potential is realized or not, because you don't measure the gene expression. You don't know that's what goes on. All right. Sorry about that distraction, but that's very important.
Momo, one more thing I want to touch on that, because I think that's really important. So it gives you the potential, doesn't necessarily offer you a solution. And then on top of that, if you do try a solution, because DNA is never changing and doesn't shift, you really can't tell whether the solution is working or not.
You cannot. Absolutely. Exactly. So yes. So that's
what Biome can do, because we're measuring RNA, and I know we typically tell our customers to retest every 6 months or so, because that's about how long it takes for the microbiome to adjust. So, we're trying our very best to give a solution by way of personalized nutrition recommendations and supplements and whatnot. But then the retest is really the gauge on how things are going, right?
Well, to me, the real gauge is, do people feel better? But yes, the test is a molecular confirmation that things are going well. Yes.
Okay, great. All right, tell us about some of the clinical research.
Yeah, I do want to reemphasize one of the points earlier that I may have not, is that, I hear so often from people, I want to feel better. I'm sick. I have all these symptoms. And then I see them, eating bad food at a party, or ordering, fast food, or saying that they have cheat days. This is not always the case, and we don't yet understand all the molecular mechanisms. But I want to caution people that, if I were to eat 3 pork ribs once a month, just one meal that contains a mammalian diet once a month, that I would be perpetually sick. I would never be healthy. And so I must not have cheat days. And while that may not be always the case, I really, really encourage people to be really strict about avoiding bad foods and to really diversify their superfoods and not have these cheat days. These cheat days are really bothering me. If someone needs a cheat day initially in order to get to that next level, go for it, but try to work towards not having cheat days. Sorry about that, Morri. That's probably derailing a little bit.
No, Momo, this is personal for you, and the most successful entrepreneurs are people who, are obsessed with their business. So I appreciate that candor very much. All right, we're moving along here. So let's move to clinical research if we can. Please tell us a little bit about, what type of clinical research we're doing at Viome, please.
Yeah. Well, yes. So we have some time to talk about that. So clinical research powers that math. So remember, we talked about nutrition 2.0 is chemistry and math. We essentially have worked out the chemistry, meaning we have very, very high quality processes in the lab. We still have a team of people whose job is to further lower the cost. So we work literally 7 days a week to reduce the costs of our testing, so that in 2 to 5 years, it'll be tenfold less for testing. I think that that's realistic. So we're still working on that. But really, where we need to make a huge improvement over the next 10 years is, remember, our goal is to completely get rid of all chronic diseases and cancers. And so we need to go specifically into chronic diseases and cancers and develop those mathematical equations that overlay on top of the chemistry that we measure. And so what have we focused so far? We focused on things that will have the biggest impact. So the first thing that we focused on at Viome from the very beginning was inflammation. We felt very strongly, and now that's obviously completely confirmed in the literature, that chronic inflammation is a significant root cause of most chronic diseases. And so early on, we focused on, okay, what are the microbial signals in the gut microbiome that are activating our immune system, and what are nutritional signals that are activating our immune system, and how can we then teach people what to eat and what to avoid, every single person, so that they minimize inflammation. So that was early days. Next, we went with glycemic control. So if most people don't know that 30% of all healthcare dollars spent today in the United States are on metabolic diseases. So type 2 diabetes and obesity and NAFLD and so on. And so we really wanted to enable people to control their blood glucose. And we realized early on that if you just follow the glycemic index, that applies to an average person. And there are, 508 people here on this call, and not a single one of those people is average. They all are different from each other, and so and different from the average, which means that we need to measure the glycemic response for every person. So back in 2017, we launched a very large study, which we actually just completed. We launched a study where in 1 year, we recruited 550 participants in the United States, and we fed them 30,000 meals. So we paid for 20,000 of those meals, and the other 10,000 they paid for, and they recorded what they ate. But we gave computers, we put continuous glucose monitors on them for 2 weeks, and we fed them these prescribed meals. And prior to that, we collected their stool sample. And so now we gave the computers exactly the content of the food they ate, exactly the content of their gut microbiome, and the glycemic response for each of those foods. And we asked the machines, hey, can you build us a mathematical equation that can actually predict prior to any measurement how this person is going to react to each one of these foods? And we were able to achieve that, and that's now published so people can read that.
So you basically took a step back. Instead of the glucose monitor being able to say, okay, I feel this way because this is what happened to my blood glucose level, you're saying we now have the ability to predict the reaction prior to them eating the food.
That's right. And that's very significant in a couple of ways. One is that people don't have to do experiments on themselves and buy a CGM and measure different things. The second thing is, very importantly, is that even if you were to test your response to, let's say, apple consumption, if you eat the apple by itself, yes, the CGM will give you that response. It will not give you a response if you eat apple combined with different kinds of foods. And we had designed in our study by a team of nutritionists 104 different meals to not only measure the response to individual foods, but the same foods in different contexts of how much protein are you consuming, how much fat you're consuming, what other carbohydrates you're consuming. So it was a very complex study design, but we were able to build this machine learned model that now every single customer of Viome benefits. And now, over the last year and a half, we actually repeated that study in Japan, exactly the same study, 550 participants. We fed them 30,000 meals. And we're now finalizing that model so that we can see if there are any specific differences for that region, so that when we open up in East Asia, we can adjust it, but we're also taking those data and combining them with what we have. So that was a really nice study where we learned the mathematical equations that we need to overlay on top of chemistry for this Nutrition 2.0 concept. Make sense?
Absolutely.
Awesome. All right. So then, in parallel, we've been working on the most prevalent sort of conditions and symptoms. And so we recruited thousands of people with depression, anxiety, IBS, and metabolic health, and analyzed their stool, blood, saliva, and did a lot of supercomputing. So we use supercomputers 24 hours a day to analyze all these data and to learn these mathematical equations that then we port over to our consumer app that people can benefit from. And so we study about 30 different conditions, and we prioritize them obviously based on their prevalence because we want to make the biggest impact as soon as possible. So we basically take in a lot of investment money, but we spend most of that money on research, not on the consumer services, because we want to refine these mathematical equations. And so what we have done so far is that we have very, very, very good mathematical equations for about 4 or 5 different sets of conditions. And we actually published a preprint on some of those. And so the way that a study would go typically is that we would basically enroll a certain number of participants in a study, and we would measure that they have a certain condition at the beginning. So we would use a clinically validated survey. So this is not something we made up. So I'll just give you an example for IBS.
Well, hold on, Momo, because I want to hit you up with something, if you don't mind.
Yeah.
I know we have to be very, call it careful. We're not diagnosing, we're not claiming that, we're curing things, this and that. And there's some sides of our business that, in the health sciences world that, we're under strict regulation and we're doing things the right way. But I want to hit you up with a question here that,
Yeah.
you tell me if you're comfortable answering. Does Viome actually work?
Yeah, yeah, exactly. So I will answer that question, and I'll explain how
And
we do.
maybe, you can tie it into this study. I'm interested to hear the results,
Yeah, yeah, exactly. So we're using very classic clinical research tools, and so we spent years enrolling participants with different conditions into studies and developing these mathematical models. We call them machine learned models, and they're basically mathematical equations that convert chemistry that we measure in the samples into recommendations. And we actually published a preprint on that so people can actually read about our description, how we do that, because that's a longer discussion. But then we asked ourselves, okay, great. We have spent tons of time, tons of money learning how this whole thing works. Does it actually work? Just like what you said. And so we enrolled participants into several clinical research studies. These are interventional studies where we enroll them and we measure a certain condition at the beginning. And so we used clinically validated surveys that clinicians use to assess the level of disease activity. And so, for example, I'll give you an example for IBS, irritable bowel syndrome. We use Rome 4 criteria. These are gold standard clinical tools that evaluate what is the level of disease at that point in time. And so we enrolled people with a certain level of that disease as measured by the Rome 4 criteria at the beginning of the study. And then we fed them the Viome diet and supplements, right? We asked them to follow these. And then after a certain amount of time, we again measured using that same clinical tool, what is their disease activity level, right? So I want to make sure that we say that we are not treating any diseases, nor do we make any claims to treat any diseases. We are just showing the changes in the clinically validated survey scores from the beginning of the trial until the end of the trial. That's what
So
we're
like an
showing.
IBS, we're looking at something like abdominal pain for, and certain symptoms. And that's what we're kind of measuring.
Exactly, exactly. So we're not diagnosing anyone with IBS. We're not doing anything with IBS, but we are using clinically validated tools that measure the level of IBS activity for a person. And we're using those at the beginning of the study and at the end of the study, the nutritional intervention. So we've done this now for 4 different conditions. So we've done it for IBS, depression, anxiety, and type 2 diabetes. And again, we're just using clinically accepted tools. We're not making any health claims. We're not treating any diseases. And we're reporting those deltas or those differences from the start of the interventional trial until the end in this publication. But basically, in summary, we have been able to achieve, or our study participants have been able to achieve very significant, somewhere in the order of 35 to 40% reduction in their clinical score within a few months by following the Viome diet. Now,
Wow.
These studies are very preliminary in a couple of ways. One way is that we did not actually collect the information from the participants as to what their level of compliance was, meaning we didn't ensure that they were 100% compliant, they didn't have cheat days and so on. So that will obviously affect the overall efficacy of the intervention. And we can do that better in the future, and we will do it better in the future, but that's a limitation of these studies so far. And the second limitation is that we didn't have a control arm. We didn't compare our nutritional intervention with a placebo control or something like that. So I just want to be upfront about that. However, we are currently planning to start running randomized control trials. These are placebo-controlled randomized control trials. We're planning to launch 4 of them this summer. And these are the gold standard. These are very much mimicking sort of the phase 3 of drug trials, except they're not going to be drug trials. They're going to be in the wellness space. We're not going to be making any health claims. We're not going to be treating any diseases, but we are going to use very similar clinical tools to assess the change in the symptoms and condition at the beginning and at the end, and compare our nutritional intervention with placebo control to see, well, how well does this work, and does it work? So we're going to test that in a really thorough way.
Amazing. All right, thanks, Momo. So, all right, well, let me switch a little bit to actual diagnostics and therapeutics. So, an organization that everybody knows and we respect and admire greatly is the Food and Drug Administration, the FDA. And relatively recently, we got their attention. And there was some interesting stuff that happened, and some interesting recognition from them. So can you tell us a little bit about what got their attention and what it means?
Yeah, so I spoke earlier about the grants program, where our systems biology platform is an open access platform. And so Queensland University of Technology from Australia wanted to work with us and develop this application, but they didn't, they're a university, they're a research institution. They're not really a commercialization arm. So we worked together to collect and analyze samples from people with oral and throat cancer, and then healthy controls. And that outcome was so good. And particularly, our saliva test was able to detect early cancer stages. So one tricky thing about oral and throat cancer, which is pretty much applicable to most cancers, is that stages 1 and 2, if detected, can actually be very effectively treated. But stages 3 and 4, which are most commonly detected, they're not treatable very well. And so our objective there was, hey, can we actually identify an RNA biomarker signature that tells us that someone has stages 1 and 2, or 2 of oral and throat cancer? And so
once we got using our same underlying technology.
Identical technology. It's basically, our saliva test is identical to our stool test. The robot doesn't even know that it's performing saliva or stool test. It's identical. And so, yes, it's exactly the same technology, but it's just collecting a saliva test, and so collecting saliva. So we stumbled upon this, and the results were extraordinary. And so we submitted this to the FDA, and the FDA loved it so much that they gave it a breakthrough designation. Now, breakthrough designation is given to a detection device if it meets certain criteria, and one of those criteria is that can literally save lives. And we showed actually health economics data that shows that if we implement this early detection for cancer in a population, that we can save many, many lives. And so that's one of the reasons the FDA liked it. Now, one of the reasons that FDA didn't like this up front was that this is the first metatranscriptomic test submitted to the FDA for the approval. So it took them a little bit to understand the technology and to understand the pluses and minuses and to be able to truly evaluate it. But once they dug deeper and found out how good it was, they accepted it. So this was the first breakthrough device designation for a metatranscriptomic technology. So we are now very, very hopeful that we will be able to develop many other tests. And in fact, we're already developing some additional tests for early detection. What's really cool about the breakthrough device designation is that we get special treatment during the device trial that we're actually starting right now to get this FDA cleared. In that normally a device trial may take something about 2 to 5 years, whereas for us, it may take only 1 year, because every timeline that the FDA normally has for other devices, it'll be shortened by 70%. So they, when they have to respond to others in 3 months, they will respond to us in 1 month. And in addition, they have a person who's dedicated to the success of this program. So they're there for us at all times. We don't need to wait for others to respond. So we are now, speaking of research, we're now pursuing what's called a pivotal trial, which is a full trial with several thousand participants that we obviously have to invest a lot of time and money into, to provide the FDA with the final results on a very large cohort to show them how well this works. And then they will either clear it or not clear it, depending on what the data say.
Is the FDA handing out breakthrough device designation something that's common?
No, it's not common. People can read about that, how tough they are. I mean, they have to satisfy certain criteria. And no, they're very tough, and they were super inquisitive and thorough in their analysis of our data. So yeah, we're very proud of that.
So in essence, our test, once it's out, can essentially diagnose oral and throat cancer before a visual diagnosis, or before certain imaging, because you're getting it at the earliest possible stage.
Yeah, so it's actually not a diagnostic tool. So diagnosis is always finalized by a doctor. Mm-hmm. So this is not a diagnostic test that obviates the need for a doctor. This is a detection test, which means that we currently, with the preliminary data that we submitted to the FDA, we have a 95% specificity and 90% sensitivity for detecting even stage 1 of cancer, which is far beyond anything that's available today, including all the visual tools and everything else. So it is far better than what's available today in clinical practice. But our test, if it's positive, will then inform the doctor to dig deeper and to find a lesion which wouldn't have been recognized otherwise, and then do the gold standard, which is pathology. And pathology means a biopsy followed by staining in a cytology lab. That's still the gold standard for final diagnosis. Maybe one day with enough data, after our pivotal trial and so on, maybe this device becomes diagnostic, but we're not there yet.
Got it. Wow. Momo, this has really been incredible. And, I've learned so much that I didn't know before. And I really, really appreciate your time here today, and, all your candor and really the transparency. And that's, makes it a very important thing for me, and why I chose to be at Viome is because, people like you are, we're so open, and it's all about moving humanity forward. Momo, before I kind of close up, any last comments, any last thoughts that you want to throw out?
The main takeaway that I would like everyone to take home is that the absolute main purpose of Viome is to serve humanity. So we did not form this company because the founders needed money or fame or anything like that. We had extraordinarily good lives before this. So we didn't need this. We are doing this, and we're working 7 days a week, 16 hours a day to really serve the humanity. So that's really the overall message. And I encourage everyone to help our mission in any possible way, it doesn't matter. There's many ways to help us out. And so, please help us on this mission, and promote us, and help us find clinicians and researchers who want to work with us and use our platform, and help us find more customers and spread the word and all of that good stuff.
I love it. Thank you so much. All right. Well, just an amazing, amazing day. Thank you so much, Momo. We don't actually profit on our kits. In fact, every time the price comes down, we pass that along to the consumer, but we're still doing our very best to make this even more affordable today. And we've been given the green light by our team to give everybody who attended this webinar today an additional code. So if you want to order another kit for yourself, your friends, your family, or you want to actually take advantage of our precision supplements, we're going to be offering this code Signal4, SIGNAL, and then the number 4. That'll get you an additional $20 off any Viome test kit, whether it's gut intelligence or health intelligence, or if you've already got your kits and you want to try the precision supplements, probiotics, prebiotics, vitamins, minerals, and supplements, that code will also give you 20% off of the current price for 3 months of a new precision supplement plan subscription. These codes and these offers are good through this Sunday, April 24th. And with that, I would like to say, on behalf of myself and the entire team at Viome, thank you to everybody for staying on for the hour. We hardly missed out on anybody here. We had a tremendous audience of hundreds of people. Thank you so much, and really appreciate all of you joining our mission in moving humanity forward and imagining a world where illness is truly optional. Thank you, everybody. Thank you, Momo, and everybody for making this possible today. We look forward to seeing you next time.
Thanks, everyone.